We're going to take a look at the cell membrane of the cells. And so we'll take a look and see what the structures of those membranes are made of and how they're going to function. So the cell membrane, or the plasma membrane, this is essentially the barrier between the cell's contents and the outside of the cell. And so this is what's going to keep everything that's inside the cells in and everything that is out of the cells out. And in order to do so, we have a membrane that is permeable to things, and so it's selectively permeable. It's only going to allow certain things in and out of the cell. This is going to maintain that structure so that we are sure of what is allowed to come into the cells, make sure that things that are potentially harmful to the cells don't get into those cells themselves. So when we look at a cell itself, and so here we have a nice spherical cell, and we can see this outer membrane made up of a phospholipid bilayer. So we have two layers of substances there in terms of the proteins, and made up of phospholipids. And so the outside of the cell, where we have all of the extracellular fluid here, is going to be water liking, and so it's going to be a lipid that is hydrophilic. And then we have a phospholipid portion here that is hydrophilic as well on the inside. So the heads are hydrophilic and the tails are going to be hydrophobic for those. And so we make sure that the hydrophilic heads are pointing, in a sense, out towards the water liquid portions, whether it is on the outside of the cell, so in the extracellular region, or whether it's in the intracellular region. We're going to have water essentially next to either side of them. The tails are hydrophobic, and so these are water disliking molecules that are there, and so they're not going to mix with water itself, and that's going to help with that permeability of the membrane itself as well. So in order for things to come through there, they're going to have to be able to be a lipid soluble substance in order to get through that layer of lipids in the center. In addition, we're going to have upwards of about 20% or so of the entire phospholipid bilayer made up of cholesterol. So we do need some cholesterol in order to maintain the integrity of those membranes. This is going to provide for some of the stability, the resiliency of the membranes themselves as they sit there in the plasma membrane. In addition to cholesterol and the phospholipids, we're then going to have an assorted number of different types of proteins that do different jobs embedded within that phospholipid membrane as well. So we have here this idea of the fluid mosaic model. And so we have essentially fluid portions that are within the membrane itself. And so that fluid portion that is there is the actual lipids. And so within that, we have these other structures that are there, these proteins that are floating within the lipids themselves. And so they're kind of floating along. Sometimes it's easiest to think about it, this is as kind of being something similar to like having a pool filled with a whole bunch of like ball pit balls. And the ball pit balls are sitting on the surface of the fluid. Those would be our proteins, and the fluid portion then being the lipids there as well. The major component of this fluid mosaic model is the phospholipids themselves, and so those are going to make up the largest component of them. We then have proteins that are embedded within it. We have our cholesterol that we mentioned before. Those different proteins are going to have different functions along the surface of the cell. We're also going to have some carbohydrate portions, and so some of those carbohydrate portions attached to the outside of the cell are going to serve different functions as far as communicating and processing things that come to the cell itself, and allow for some components there that's attaching to it. And then we have other things that are going to help with the stability of the cell itself, which are things like the cytoskeleton. Those cytoskeletons are going to allow us to essentially maintain the structure of the cell itself on the inside of the cell. So here we can see kind of a generalized idea of that phospholipid bilayer. We have our phospholipids, obviously, with the hydrophobic tails and the hydrophilic heads on the outside, kind of pointing towards the water portions on either one. We've got all kinds of different kinds of proteins that are there. Some of these are what we call integral proteins. They're actually going through the entire membrane. Some of them are going to be just on the surface. They're going to be peripheral types of proteins. So those peripheral proteins may only lie within the inside of the cell, or they could come through to the outside of the cell as well, but they're just along the edge of the peripheral portion of it. Here we have part of that carbohydrate portion. So here we have what we call a glycocalyx. This is going to be part of our cell-to-cell signaling components that are there. A little bit of the cytoskeleton, and that embedding into the walls and allowing us to maintain the shape and integrity of the cells themselves. So some of those proteins do different things for the cells. And so one of them is that it allows for transport. So we do have to be able to get certain things in and out of the cell. And so here we have a transport type of a protein. This is going to allow for specific substances to come in and out of the cell. Sometimes it's going to be kind of open like this one. Other times it's going to be very selective in terms of what's there. In the next video, we'll talk about those different proteins and how they actually function within the membranes themselves there as well. But either way, they're either going to allow something into the cell or potentially allow something out of the cell along with the movement that's there. Other times we may have receptors, and so this receptor here is going to receive some sort of chemical that comes along. So in this particular case, it happens to be a ligand, and so we have something like a neurotransmitter or a hormone, some sort of chemical that is binding to the outside of that receptor, and that's going to potentially make a change on the inside of the cell. And so it may change the receptor itself, it may change the functioning of the cell inside, may send another signal along into the inside of the cell. It all just depends on what that receptor itself is. We then have identity markers. These are things like our glycocalyx. And so our glycocalyx is made up of a combination of proteins and carbohydrates, and that allows for signaling between cells. So if something like say a white blood cell comes along here, it's going to be looking for the right type of glycocalyx to be there, essentially saying this is the cell that's supposed to be in the body. These are the kinds of cells that are there. Don't bother us, don't harm us. And so that actual binding site, if the binding site came along, it would in a sense make the correct attachment there. So it would have to have the correct type of protein there and carbohydrate combination in order for that immune cell to ignore it and kind of move on its way. Other times we have enzymes, and so enzymes are going to be able to catalyze and speed up chemical reactions that may be occurring both either in or out of the cell itself, and that's going to allow us to speed up processes. It'll also allow for signaling within cells and between cells. Many different processes are going to utilize that enzymatic activity that it may have there. From there, we have things like anchoring proteins. And so this is going to allow us things like our desmosomes, allow us to anchor between two cells. And so part of the cytoskeleton proteins are going to extend out for that, things like intermediate filaments, and allow us to maintain the integrity of the cell itself again, so that we maintain the size and the shape of the cell as it is there. Not only do those anchoring types of proteins allow us to maintain the cell shape, we can also have other types of proteins that are actually going to help to hold two cells together. And so this one here is almost as if we were binding or welding those two cells together in a sense. And so this is going to allow for that cell adhesion there in between there. There may or may not be really any kind of space in between those two cells. In some cases, it may allow for a certain amount of interstitial or extracellular fluid to move in between the two cells. At other times, those anchoring proteins may actually make it so there's basically no space in between them. And so it's going to kind of bind the two cells together to ensure that they're not able to move anywhere. The lipid bilayer itself gives the membrane its flexibility and its fluid characteristics that are within the cell that are there. If that cell should happen to be penetrated it has the ability to kind of seal itself off. And so as long as the damage that has occurred there is not too great, it has that ability to kind of seal itself there. Sometimes it uses this process in order to actually move things in and out of the cells. And so we can have exocytosis and we can have endocytosis. And so exocytosis is going to allow us to eliminate substances from inside our cell. And so we have this little vesicle here. We have some sort of container, in a sense, inside there. And perhaps there's some content that's inside there that we want to get rid of. And that little vesicle then is going to move to the surface of the cell. It's going to open up and allow that contents to leak out. And so we're going to do exocytosis in that way. We can do endocytosis in basically the reverse. And so here we have our cell, and then here we have a little vesicle that needs to get in, or some substances that need to get into the inside of the cell. And that cell then, in a sense, opens itself up, and then it eventually brings it into the inside of the cell. And maybe we have some orange content inside there. And then that's now consumed into the inside of the cell itself. And so we're bringing substances into that cell that we may need. Sometimes that vesicle then may travel through, go to kind of like the other side of the cell, and then it exocytoses it out onto the other side. And then those substances can then go across a membrane, something like that. So endocytosis, exocytosis, just a graphic representation here. Once again, exocytosis is allowing for those substances to move out of the cell. Endocytosis is bringing that substance into the inside of the cell. So it's going to be brought in. This could be digestive compounds. It could be something that we're going to absorb into the body, and it's going to go all the way through the cell and then end up going through exocytosis on the other side. That's there. And so that allows us to move those substances across to the other side of the system itself. All of which is going to basically follow a concentration gradient, which is what we have up at the top. And we'll get into concentration gradients in more depth in the next talk.